Good afternoon, everyone. As Soha mentioned, I'm Nidhi Nandu, a PhD candidate in the Yigit Research Lab in the Chemistry Department at U Albany. Here, I work with nanoparticles and DNA to design nanobiosensors. Most of us encounter nanoparticles in some form in our daily lives cell phones, nail paints, sunscreens. What makes them interesting is that the nanoparticles showcase different properties at their smaller scale as compared to when they are larger. DNA, on the other hand, is a very versatile molecule which not only interacts with DNA, but also other molecules like proteins and even large cells. One of the common designs for nanosensors is to use DNA sequences programmed specifically to interact with a target. When the target is introduced to this nanosensor, the DNA interacts with the target and we get a signal which has lots of information about the target. When I say programming DNA sequence, I mean that the sensor would have a DNA sequence to detect the young man in the painting on the slide and a different DNA sequence for the hut in the painting. Basically, a new DNA sequence for each element of the target. However, we often see some background signal or noise due to off target detection. That means the sensor with the sequence specific for the hut would give some signal even when the hut is not present. Researchers often see this as disadvantageous and try to suppress this noise to make the sensors more sensitive. I decided to look at this noise a little differently and see if I could get some information from this noise. To do that, I developed a non-specific sensor. Instead of using specific DNA sequences for individual elements, I used a few common DNA sequences along with different nanoparticles. When any target is introduced to this nanosensor, the recovered signal is due to a combination of all the elements of the target. I looked at the signal patterns obtained with the help of artificial intelligence. The sensor I designed could pick up minute differences in the samples and differentiate them. But how can this help us? Imagine a scientist working with cells and not realizing that the cells are mutated till it's too late. The experiments could be compromised, and this has already resulted in many retractions of publications. Being able to detect even the smallest changes in the cells could have prevented this. My sensor has already been able to differentiate these uh, differences in breast cancer cells. I have been able to differentiate even between different types of milk whole milk, soy milk, cashew milk. And all of this done is used by using the same set of nanoparticles and the same DNA sequences. So thank you.